Now that we've discussed the cell cycle and mitosis itself, let's talk a little bit about how the cell cycle is controlled. There are in cells a series of checkpoints, and we'll talk about how that works in terms of the control. There are different rates that apply to different kinds of cells. Different cells divide and reproduce at different rates. There are various things that are worked toward controlling cell division, one of which is the presence of the particular nutrients that are needed by that kind of cell. Also, there are growth factors, which are proteins that stimulate cell division when necessary. There is something called density-dependent inhibition, in which once the cells get crowded into the area that's available for them, they'll stop dividing. And then finally, anchorage dependence, which is the need for cells to be in contact with a solid surface in order to divide. If you culture cells in liquid, they don't really grow very much, okay? They'll survive, but they don't necessarily grow very much. When you put them in a culture medium here, they'll still stay about the same, but once you add the growth factor, that's when cell division starts to begin. And so there's something in the growth factor that stimulates that cell division. Also, they won't really grow in a liquid, but if you put them in a solid surface that they can anchor to, then they'll grow and form a single layer of cells. Once they fill that surface, that's called density-dependent inhibition. Once they're filled up, they'll stop growing until there's a gap or a place that needs to be refilled, and then cell division will fill the place where they've been removed until you get that single layer of cells there, and then they'll stop dividing again. So there's definitely something about the lack of a solid surface and the lack of crowding that encourages a lack of solid surface that discourages growth, and a presence of crowding which discourages growth as well. The cell cycle control system is a series of molecules that are circling or cycling in the cell, and once these factors come together, that will trigger the cell cycle and coordinate the events of the cell cycle. There are several checkpoints in the cell cycle that either can stop an event or signal an event to proceed depending on what's happening and what that checkpoint requires. There are three major checkpoints in the cell cycle, are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. And so when you look at the G1 checkpoint, that's the point where the surface area to volume ratio could be a critical factor, as well as the presence of an open space to divide into, the presence of the particular growth factors, okay? And what that's going to do is it's going to say, okay, it's time to go ahead and start entering the S phase of the cell cycle. Let's start replicating the DNA and get ready to divide. Something causes the cell to leave that cycle, or just the G1 cycle, G1 place. And sometimes there's some cells that enter a non-dividing G0 phase. So when they're crowded together, or when there are certain kinds of cells that have different kinds of growth factors involved with them, they end up in this non-dividing G0 phase, and they'll stay there for a long time until it's necessary for them to divide. The G2 checkpoint checks to make sure that the DNA is properly copied and that all the other molecules that are necessary are ready to go into the M phase, into mitosis. And finally, the M checkpoint makes sure that all the chromosomes are attached to spindle fibers and things like that, and it's ready to continue with division. It's basically in metaphase that's decided. So this is very important, as you can understand. There's a lot of research into control of the cell cycle. This is one of the hottest areas in biology today, as you can imagine. It definitely is something that's important to understand when you're studying how to prevent or how to treat cancer cells, cancer tumors, and so it's a very, very hot topic today in biology. Here's a diagram that shows you basically where these things occur. The G1 checkpoint is close to the end of the G1 phase of the cell cycle. Right at the end of the G2 phase, we're checking to make sure everything is ready to go. And then finally in the M phase, the M checkpoint is making sure that all the chromosomes are attached and everything is ready to divide the cell into two. Now, when you have cells that grow out of control, that's what produces cancer. Cancer occurs when you have cells that escape those controls of the cell cycle. for some reason something is making them grow uncontrollably, whether it's one of the cell cycle proteins, whether it is some other factor that's turned on or turned off. There are various things that can cause it, and this is also a large area of research today, as you can imagine. The cancer cells, you know they're cancer cells when they divide rapidly, often when there's not even a growth factor present, when they spread to other tissues through the circulatory system, and when they grow without being inhibited by other cells. In other words, instead of forming that flat layer in that culture plate, they'll actually grow up in mounds, and that's what causes something called a tumor. A tumor is an abnormally growing mass of body cells, and there are various kinds of tumors. Some are cancerous, some are not. Benign tumors are ones that remain at the original site. They may or may not be, they're not cancerous because they're not invading other tissues, but malignant tumors are going to spread to other locations. This is called metastasis, and it occurs with cancer. That's what a malignancy is. Here we have a tumor starting in the tissue here. This is glandular tissue in the breast, and you have a tumor beginning there. And as long as it stays right there and doesn't grow anymore, then that could just be a benign tumor. Once it starts invading other tissues, that's when you know for sure that you have a cancerous tumor. And then finally, when it gets into blood vessels and lymphatic vessels and migrates to other parts of the body, that is called metastasis. Cancer cells are, cancers are named according to where they originate, okay? And there are three main kinds of cancers. There are carcinomas, which are in body coverings, either inside or outside the body. Sarcomas arise in supportive and connective tissue, like in bones and so forth. And then leukemias and lymphomas arise from the blood-forming tissues. Now, cancer treatments there are various kinds of treatments, and again, there's a lot of research going on about what's most effective, and it depends on the particular kind of cancer and what stage it's in. But generally, localized tumors can be removed surgically and or treated with radiation, sometimes localized chemotherapy. Metastatic tumors must be treated with some kind of chemotherapy because it's kind of like a hunter-killer drone that can hunt down and kill the bad cells before they have a chance to spread any further. There are numerous other things, and we'll watch some videos and have some discussions about some of these things in class. This concludes the notes for Unit 7.